Okay, coming on back to our worksheet here, because that's where I can actually do this stuff. Here is that polypeptide. And what we're going to do, as we said, is circle some hydrogen bonding donors and acceptors in the backbone. So bear in mind, there are a couple of hydrogen bonding donors and acceptors in the side chains. We're going to ignore them for now. Although if you did circle them, congratulations if you identified this as being an acceptor in the N and then a donor from that H, or an acceptor with this O and a donor for that H. As far as what's in the backbone, though, what we see is a very repetitive thing. We'll notice that each of these O's right here is a hydrogen bonding acceptor. And in fact, for that matter, so are the N's right here. But of course, if I want to start making some hydrogen bonding attractions, just simply having hydrogen bonding acceptors around isn't enough. I need there to be some donors. And so the donors, I told you before, the H that was still attached to these N's would be important. That's what these guys are. And so those are my H bonding donors. So we've got a little of both of those going on, and it's a very repetitive thing. When we start into secondary structure, what you're going to observe chiefly is that it's interactions between these acceptors and these donors. The nitrogens themselves don't get terribly involved because they can't interact with these donors for the simple fact that they're attached to them already. So that's those. Moving on to the identification of the termini, which I did not put on the slide, but we'll go ahead and do that right now. Pause the video if you'd like, and identify, have I written this N to C or C to N? Okay, hopefully what you see is on this side, I've still got a nitrogen loose, so that's my N terminus. Over here, I've got the carboxylic acid still loose, that's my C terminus. And in general, when we call out a sequence, we're going to build them that way, okay? Now, time for some classification. Let's go ahead and bring that up on the PowerPoint. First off, naming. So I hope what you came up with here is that this is proline. This one right here with this giant side chain there with all the aromaticity going on is tryptophan. Moving over here, we've got the single primary alcohol, that's serine. Here's valine, which we already saw once before. And here's cysteine, which we already saw once before. And so if I'm looking at abbreviations and both the one letters and the the 3 letters and the 1 letter abbreviations, this is what we've got. Proline, PRO, tryptophan, TRP, serine, SER, valine, VAL, cysteine, CYS. A lot of them are going to have abbreviations that are just simply the first 3 letters of their names. The only ones that don't, tryptophan you'll notice doesn't, and part of that is because otherwise it would look a lot like tyrosine, which is TYR. If you had one that was TYR and one that was TRY, you could imagine people might get themselves confused. One of the only other spots where you see this kind of get interrupted is on asparagine and glutamine. Notice that those are not ASP and GLU, and the reason for that is that those abbreviations are already taken. ASP is aspartic acid or aspartate. GLU is glutamic acid or glutamate. Asparagine and glutamine, slightly different. They end up getting it with the N at the end of them. As far as the others, I think every other single one of these besides them and tryptophan, the 3-letter abbreviation is pretty straightforward. You will see where there are multiple amino acids that have the same starting letter, you always have one that gets the first letter of its name as its symbol, and then one that gets something else. So for instance, you've got threonine, tryptophan, and tyrosine. They can't all be T, so tryptophan is W, tyrosine is Y. Phenylalanine and proline can't both be P, but phenylalanine starts with a F sound, so they go ahead and make that F. Leucine and lysine can't start with the same letter. They can't both be L, so lysine just moves up in the alphabet to K. And then same deal again with the glutamine, the glutamic acid, glutamine, and glycine. Glycine gets the G. The 2 acidic amino acids get D and E, because additionally we have asparagine and arginine and alanine up here. Glutamine gets Q, because that's almost all that's left. Asparagine gets N. Arginine gets R, so it kind of sounds pirate-like. But those abbreviations are generally going to be there in some way to try to help you with this and tell them apart from each other. Oop, wrong way. Here we go. So that's them, and if we go down the line of classifying them, you'll notice that what we've really got here are a pair of polar amino acids in serine and cysteine. Valine is all alkane. Tryptophan, despite this one hydrogen bonding group, also has a ton of carbon, so it tends to be classified as nonpolar, and so is proline. So 2 polar amino acids, 3 nonpolars in the midst of that. Now, the first level of actual structure that we get once this thing is put together is that when you have a large molecule with polar and nonpolar regions, it has a tendency to begin folding in on itself. And so what we're going to see here after we've got this first level of structure is that these secondary structures are going to form to start dealing with the hydrogen bonding groups on the backbone. Wrong direction. Here we go. These hydrogen bonding groups on our backbone, the green acceptors and the maroon donors, want to be satisfied. They attract each other, and so our first 2 levels are going to involve that. There are certain groups, certain amino acids that are more amenable to some of these structures than the other. Particularly proline, we're going to see, only does one of them particularly well. But the 2 primary ways this happens are by what we call, in satisfying this sort of thing, are by alpha helix formation. So in the case of an alpha helix, what you've got is the backbone essentially wrapping itself around a pole, very much the same way as the DNA double helix that you see. So it's got a central axis, and as it wraps around, what you've got are the carbonyls from, for instance, this bottom one pointing up, the NH groups from this next one up pointing down, and they're able to form an attraction with each other. And since everything's got the same backbone, this is a very common structure. Now, what happens when this occurs? I'm going to take my other piece of this here and come out to the straight ahead camera, is that when I've made this structure, what I've essentially done is I've taken the backbone, which was like this, and I've wrapped it around like that. And I know that in this little cylinder, I've got all of my carbonyl and amide groups basically pointing along this curvature, right? And so remember the way that tetrahedrons work. If you've got 2 things that are occupying a specific plane, the other 2 things have to be pointing away from that plane in either direction, kind of not quite perpendicular to it. It's going to be a 109-degree angle, but definitely into and out of that plane. So if the plane of the backbone is this cylinder, then the hydrogens are pointing into the cylinder and the side chains are pointing out. And so what I wind up with when this thing folds is I satisfy a lot of hydrogen bonding verve on the backbone, and I wind up with all of my R groups pointing out along that cylinder. And so I can do interesting things with this if I want an alpha helix to be for maybe amphoteric, or what's the word I'm looking for? Amphipathic. Sorry, wrong word. Amphipathic. Because what I could do is build my protein such that one side of it is all nonpolar amino acids and the other side is all polar amino acids. And I might get some interesting positioning as a result of that, based on making one side of it very hydrophobic and one side very hydrophilic. Similarly, I can take these sorts of things, and instead of wrapping them around a central axis, I can do the thing that proline tends to really help out with, which is I can turn it into an anti-parallel sheet. So in other words, I've got one strand running this way, it hits a little hairpin at some point, and the next strand comes along that way. Hairpin comes along like this. And bit by bit, the backbone now forms a sheet. And whoops, that's not what I wanted to do. I wanted to do that. Ooh, did I zoom in by mistake? Probably not. Anyhow, in that case, now what my backbone is doing is it's running back and forth along a flat surface. And so if I have that, again, remember what we said, the H's and the side chains are going to be going into the sheet and out of the sheet. And so again, this time around, because I'm wrapping in different directions, and based on what we saw, for instance, when we did this guy a little while ago, where I end up with things going, side chains going up, then down, then up, then down, the end result is that when I have a beta sheet, what I tend to see are R groups alternating back and forth. And again, I could do the same thing. I could have side chains on one side of the sheet be very polar, very charged, very happy to interact with water. And I could have R groups on the bottom of the sheet be very nonpolar, very hydrophobic, very oily. And I might get that to sit in a very specific position as a result. As I said, less flexible amino acids like proline tend to help out with this because you can't get the curvature that's necessary to make the alpha helix. Now, like we said, these two structures tie up the backbone in hydrogen bonding, but they leave the side chains exposed to the solvent. And so what we're going to look at in our next video are the forces that come up between the side chains that give rise to what we call tertiary structure. I'll see you for that video next time you click.